Hi guys. First, thanks to Jim and the other organizer for arranging this meeting. I have been looking forward to the event, and it's a little sad that we were not able to travel this time. But hopefully soon there will be new opportunities where we'll love to travel and meet again in person. Anyway, today I'll present some of our research where we investigate anabolic steroid use and long-term effects on the brain. So this is an overview of the studies that I have initiated. And our main study is the first one, the brain and behavior study. And that is also the one that I will focus on today. And in this study, we have collected data throughout many years with 2 major data collection time points. In total, we have had 140 unique steroid users and 110 weightlifting controls that have participated in this study. Some of these participants have also participated twice with 3.5 years between the time points, giving us a longitudinal aspect to the dataset. And while our main focus is brain imaging, cognition, and mental health, you know that anabolic steroids is associated with a diversity of side effects. So from the second time point, we also broaden our collaboration to include researchers from different disciplines. So at that time point, we also have had a cardiac examination, blood vessel examination, and we have sampled blood. So we have detailed levels of hormones and other biomarkers that we believe could be related to brain health. And yeah, this is a picture also for where we work, where we spend our days. And this is a recent picture of the core research group. So when steroids are used in order to increase muscle mass, typically high doses are taken over longer periods of time. And we aim in our research to see how such long-term high-dose use affects the brain, cognitive function, and mental health, and how it affects brain health in old age. So we know that anabolic steroids may cause psychological and behavioral side effects. So then we also know that it affects the brain. Here is a word cloud of the most experienced psychological side effects from our recent study of 100 AAS users. And some of these side effects likely reflects temporal hormonal fluctuations, while others could reflect more permanent changes. I would like you to have in mind that there are different ways that AAS might affect the brain, and this is very similar to the effect of other drugs of abuse. We have temporal activational effects. For instance, AAS-induced mood swings is likely a consequence of hormones being present or absent, binding to their receptors in brain regions implicated in emotional processing. It could also be indirectly by influencing the signaling of neurotransmitters in the same regions. But substances of abuse and AAS might also change the structure of the brain and induce long-term alterations in behavior. And I like to think of memory impairments as one such long-term brain alteration. In support of structural brain changes of AAS use, several cell culture studies have shown that high-dose AAS use may cause neurotoxicity, death to the nerve cells. And there's also animal models and a handful of human studies that have shown that AAS use is associated with impaired cognitive performance. We also know that there are medical side effects associated with AAS use that may impair brain health, for instance, hypogonadism and a variety of cardiovascular risk factors. So one of our main research questions is to see if long-term use of steroids impact brain structure. And to do that, we have recruited male steroid users with more than 1 year of cumulative steroid exposure, as we're looking at long-term effects, and we have compared them to a group that are similar in many ways, that they are matched on heavy resistant training. So we have a weightlifting control group. And all the participants, they scan their brain, and we compare the volumes of different brain regions, and we compare the regional thickness of the cerebral cortex, which is the outermost layer surrounding the whole cerebrum. So the MRI findings were that compared to the weightlifting control group, the steroid users, they had smaller neuroanatomical volumes. And smaller volumes were found for total gray matter volume, cerebral cortex volume, putamen, which is a large structure of the basal ganglia, corpus callosum, which is the fiber bundle tract connecting the two hemispheres, and the clearest findings that also survived correcting for when we kind of did analysis where we left out those with other substance use issues were the ones for the larger volumes, total gray matter volume and cerebral cortex volume. And you can see that I've written also note that there were no differences in intracranial volume, and that is kind of important also to notice, as the intracranial volume is the volume that surrounds the brain, and it's kind of a measure for how large the brain, it says something about how large the brain it was supposed to be at its maximum. And if there were differences in intracranial volume, then we could have suggested that there were differences in brain volumes prior to steroid exposure. But now we do not have any indices that suggest that there should be differences in brain volume prior to exposure. And here are the findings visualizing regional cortical thickness differences between AAS users and control. And the findings are illustrated on lateral and medial surfaces on the brain, right and left side. So that's why you have kind of 4 pictures for visualizing these effects. And blue color illustrates areas where the cortex is significantly thinner in steroid users compared to weightlifting controls. And you can see that it's kind of widespread effects. And in the lower picture, we have kind of looked at those with the longer steroid exposure, those with more than 10 years, and compare them to weightlifting control. And you can see then that longer exposure is associated with even more widespread effects. Are these differences related to cognitive performance in our sample? And to do so, we have investigated cognitive performance using a broad range of cognitive tests, and then we have grouped these tests into meaningful cognitive domains. And the main domains that we have looked at is working memory, speed, learning and memory, fine motor speed, problem solving, and executive function. And in all analysis, we we adjust for age and education, as we know that they are factors that always are related to cognitive performance. So we remove the variance caused by that. And so in this analysis, we can see that there are differences between the groups on almost all cognitive factors that we have investigated. But there could also be other things that could influence cognitive performance. For instance, premorbid abilities and other use of substances of abuse. So we also have conducted analysis where we try to remove the variance caused by that, to see that it's not alcohol and cocaine use that causes the differences. And when the findings were kind of stable also when we do that, and the most prominent findings are seen for working memory, speed of processing, and problem solving. And we've also seen for memory performance that there is dose response effect. So we did comparison within the steroid group, comparing steroid users with shorter exposure, less than 5 years, to those with a more prolonged exposure, more than 10 years. And it was particularly for memory performance that was more impaired in those with a longer exposure. And 36% of our sample also reported memory problems to be a side effect of use, and increasing to 50% of those that had used steroids for more than 10 years. So based upon our structural neuroimaging data, we have now estimated the biological brain age of our participants, which is kind of cool. Brain age have been calculating using artificial intelligence, machine learning models, where a large lifespan dataset of healthy males are trained to make a model of how a typical brain looks in a particular age. And then we apply this model to our sample, and then we get the brain age of each participant. And the difference between the predicted brain age and chronological age is termed the brain age gap. And it can tell us if the aging trajectories are deviant from the population norm. And we found that AAS users, they had higher brain age gap compared to non-using weightlifters, thus could suggest earlier brain aging. Steroid dependence is a particular focus of our research group. It's quite clear that users who develop an addiction to steroids are a particular vulnerable group in many ways. They are those who experience the most medical and psychological side effect, but that continues to use steroids despite of that. So one major aim of our research group is to find characteristic traits or markers putting people at risk for developing steroid dependence. Addiction is a brain disease. Drugs of abuse, they hijack the brain reward system, and long-term exposure to various drugs of abuse, including anabolic steroids, it seems, may cause structural brain changes. So we wanted to see if there were structural brain characteristics that were associated with AAS dependence. And to do this, we compared the brain scans of dependent AAS users to non-dependent AAS users. And the blue areas illustrate areas with thinner cortex in dependent steroid users. So all the findings were in that direction. And dependent steroid users, they had thinner cortex in several regions, including the prefrontal region, temporal regions, parietal, and also in occipital regions. But I would like you to pay a little extra attention of some of the most profound findings in the frontal regions here and the orbitofrontal regions, which is areas that are critical for impulse control and emotional processing. And the findings are actually kind of similar to what have been seen for other dependencies. And it could be that this thinning in frontal regions and reduced inhibitory control could be one explanation for the kind of maladaptive behavior of exaggerated drug use despite of adverse consequences. We also looked if steroid dependence was associated with earlier brain aging. So here is the brain age findings comparing non-users, non-dependent steroid users to steroid-dependent users. And what we found was that it was actually the steroid-dependent users where the brain age gap was increased, whereas non-dependent steroid users were no different from weightlifting control group. So we have also started to disentangle factors that might be associated with our brain age findings. And as I previously mentioned, we have a very rich data set now with a lot of interdisciplinary data on steroid users and weightlifting controls. And now we're working with this data to kind of see the broader picture. And one of these things that we try to look at is if neuroinflammation or inflammatory markers are increased by or are affected by steroid use, if steroid use influences the immune system functioning, and to see how this relates to brain aging. So in normal condition, inflammation is a kind of protective effect. But chronic inflammation and in aging and aging-related illnesses, the inflammatory system is kind of in the high gear, and we get too much of it, which might damage the nerve cells and increase brain aging. So we're trying to look at different inflammatory markers and how they relate to our brain findings. And here is one interesting factor, interleukin 8, which we first see that it is higher in the steroid user group, suggesting that steroid use could have some impact on the immune system and cause this heightened level of neuroinflammation. And what was also interesting was that this factor in particular is clearly linked to our brain age gap findings. And this was true for many brain regions. And here I show you a profound region from finding from the brain age gap in the temporal lobe. And here you can also see that it was the steroid user group, and that kind of that it was this relationship was much more profound in the steroid user group. One factor that I am extremely interested in is the growth factor, brain-derived neurotrophic factor, or BDNF for short. BDNF is a protein that helps our nerves grow and are important in neuronal survival and synaptic plasticity, and in promoting learning and memory processes. BDNF levels have also been highly linked to a variety of mental illnesses, depression in particular, where love levels are seen in depressed patients and also in other disorders. And we find kind of huge differences between the groups in BDNF levels, with lower levels in steroid users. And note that this is also true for former AAS users, suggesting long-term reduced neuroplasticity after steroid use. And I think this finding is a little bit interesting also, as we know that former AAS users are a vulnerable group. They are vulnerable to long-term psychiatric illness, and it could be that reduced neuroplasticity could serve as an explanation to why that is. So to wrap it all out, we have now seen that long-term high-dose AAS use is associated with apparent aging of the brain compared to a weightlifting control group. We have seen that steroid users score lower on a variety of cognitive domains. And we have seen, when it comes to the brain findings, we have seen that steroid dependence is associated with prefrontal thinning, which resembles finding from other dependencies, and that it was dependent steroid users in particular that had this increased brain age gap. And I'll also show you some preliminary findings that I find very interesting, that steroid use might also have impacts on immune system function and neuroplasticity. And finally, thanks to all that have contributed, all muscles and brain, the participants that have used so much time to take part in our research, and a lot of these findings related to steroid dependence. You can find that in the thesis work of Lisa Hauger that defended her thesis in May. And now we have some new collaborators, new members of our research group, and we will continue to work heavily on these findings. And thanks a lot to Jim and the other organizers.